Hello everyone, welcome to T1 Academy. Friends, today in the topic of Science and Technology Rapid Revision Program, we'll be focusing ourselves with the topic of biotechnology. When it comes to the point of UPSC CSC Prelims, this topic has highest weightage. Now, how to crack this topic is also very important. That's what we are going to do today. When I was teaching you science and technology in all the previous classes, one important point that I have asked you to remember is science has unlimited capability and it can do anything and everything except few things, and knowing what are these exceptions will make you a sense. Knowing the exceptions of science, because you cannot create a life from scratch in science. You can create already existing life or you can create a modification, but you cannot create some new life. That's what it is, the incapability of biotechnology. So knowing such incapabilities, what it cannot do, is very important. Apart from that, what you are going to learn in this session with respect to biotechnology also makes a lot of sense. Number one, what you are going to learn in this session? You are going to learn about biotechnology. In this, you are going to learn about the different types of biotechnology. You will be learning what is DNA and RNA. And you will see the applications of biotechnology in various fields. You will see latest applications, and you will also be able to learn about different tools in biotechnology. Different tools in biotechnology. Basically gene editing tools, which is a groundbreaking innovation in the field of science and technology. You will also learn about technologies like CRISPR-Cas9. Okay. You will also recombinant DNA technology, rDNA technology. And you will also be learning what is a genome and Genome India project. Apart from that, you will also be learning the latest news in the field of biotechnology, like three-parent baby. What is this three-parent baby? All these topics. Apart from that, I will also make you understand about other topics which are not mentioned in these slides. Those include cloning, tissue culture. I will also be telling you xenotransplantation. Okay, and also few points about environmental application of biotechnology. All this we'll be learning in this session. So let's start first by understanding what are the different types of biotechnology, and much prior to that, what exactly is the definition of biotechnology. My friend, biotechnology stands for using biological resources in the various fields of science in order to give solutions to the problems that human has. And everyone can have their own definition to this biotechnology. But simply, when you are using a living organism, say it could be a microorganism like bacteria and virus, say it could be a living organism such as plant or an animal or any other living organism, for the purpose of solving your human problems, that type of technology is called as biotechnology. It is a use of biological processes, organisms or systems to manufacture products intended to improve the quality of human life. Take the case of insulin. Insulin that is basically used as a treatment for the diabetes patients. It is basically produced from bacteria as of today. Earlier it was extracted from the animal, but today we are extracting it from E. coli bacteria, okay, in a large scale. So such technologies have been developed in order to make the human life to go easier and easier. Now next, what are the different types of biotechnology? In general, if you want to learn about the different types of biotechnology, number one is green biotechnology. When you apply this biotechnology thought processes or techniques into the field of agriculture. Okay, when you take it to the field of agriculture, forestry, something which is related to plants or trees, then that type of biotechnology is called as green biotechnology. Now, where does this green biotechnology, or what are the different applications of green biotechnology? If you see, one, you are producing genetically modified crops. In India, there are only two genetically modified crops, BT cotton and BT mustard. What is the genetic modification? Under the genetic modification, we will try to introduce certain traits into any particular crop so that the crop can stand for longer or crop can become nutrient. For example, we have also developed a type of rice called golden rice. What is this golden rice? Golden rice is a variety of rice which was genetically modified to have highest amount of vitamin A. Highest amount of vitamin A. Now what you are doing, you are fortifying the food by using this biotechnology. What is possible? What is introduction of vitamins into the food is called as food fortification. Okay, whereas in the case of BT, you have introduced a bacterium in them. Bacterium which provides a protection to this particular crop against any other pests coming from outside. This bacterium, basically Bacterium thuringiensis, protects from pest attacks. So what do these two examples tell you in the field of application of biotechnology? One, we can provide genetically modified crops which are basically pest resistant. And we can also provide genetically modified crops which are nutrient rich, and these nutrient rich crops could help to reduce malnourishment that India is struggling over the decades, right? This will do what? Reduce malnutrition. Apart from all this, my friends, let me also tell you a wonderful thing about the application of biotechnology in the field of green biotechnology. Here, my friend, you can also develop some particular plants which can absorb the pollutants from the soil, from the water and from the air also, so that you can remove these harmful pollutants easily. So they all get accumulated into this plant body. Say you can say it roots, its trunk, its leaves, its branches, etc. And when you process this plant, a particular plant, you can get all those pollutants. Some of the pollutants like heavy metal pollutants, when you are able to extract these heavy metals, they can be used in manufacturing of various items across the world. Okay, now what can you do? You can develop, okay, organisms which can eat oil in case of oil spills, and other thing, you can develop plants which can absorb pollution. Okay, these are very important points when it comes to your exam. So that's what I was trying to produce you. Okay, we can develop good plants which can give high quantity yields and quality yields. This can be done by implanting foreign genes into the plants for economic important species. That's what the Bacterium thuringiensis. A foreign bacteria has been introduced into the plant DNA. See, here is a catch. You have seen, I can take a particular gene of a organism which is a bacterial organism and take that bacterial DNA and put it into a plant DNA. If someone asks you that an organism DNA could be inserted into a plant DNA, or tomorrow if someone asks you a plant DNA could be inserted into a human, is it possible or not? Please do comment in the comment section. Question could come like this. Apart from that, another important technology in the field of green biotechnology is tissue culture. What is this tissue culture? Tissue culture is ability to produce a complete plant from any particular part of a plant. For example, if you get a small leaf of a one particular plant, your ability to create a complete plant from that one leaf, or your ability to produce a complete plant from a seed of a plant, your ability to produce a complete plant from a stem of a plant is called tissue culture. Basically, why do we use tissue culture? My friends, there are good valuable plants across the world. They take lot of time to germinate or lot of time to grow big or lot of time to propagate themselves. In order to help these plants to propagate longer and longer areas, okay, to spread and to grow faster, because few plants take years to at least bud out from the soil with roots. So that in order to make these plants to grow faster, we use a technology called as tissue culture. Okay, in tissue culture there is an important term called totipotency. This is most important term you have to remember with respect to tissue culture. Totipotency is the ability of a particular plant cell to develop into any of the plant cell. If you want to become a fruit cell, you can become a fruit cell. If you want to become a root cell, you can become a root. If you want to become a leaf, you can become a leaf. If you want to become a branch, you can become a branch. If you want to become a stem, that is called, you can also become a stem. The ability of a cell of a plant to be grown into any of the part of a plant is called as totipotency. To develop into any cell. Okay, so this is what the application of biotechnology in the field of green biotechnology, that is application in the field of plants and greenery, is called as green biotechnology. Next, red biotechnology, that is application of biotechnology in the field of medicine. Basically, you are going to treat people. It is concerned with the discovery and development of innovative drugs and treatment. What does red biotechnology stands for? Developing new drugs and treatments. Now, how or what are the different treatments you have? See, today in the world, we are trying to provide you gene therapy, stem cell therapy, and a lot of genetic testing, which can even predict what type of diseases you could get over a period of time in the future. Now, let me try to tell you few of these important techniques. Number one, we'll try to understand the stem cell therapy. Okay, stem cells are basically specialized cells in the human body. Okay, your whole human body will consist of stem cells. Okay, what are these stem cells? They are specialized. Okay, if you want to divide these stem cells, they can be divided into various types. But let me tell you two types. One, embryonic stem cells and adult cells. My friends, how do a human born or how does any organism get birth? When female and male sexual partners, they do sexual reproduction, the sperm and the egg, they get fertilized, and from this fertilized egg, slowly there happens mitosis and meiosis, and slowly that whole cells get divided, and each of a particular cell will make and each of a particular part of your body. There is a cell for the bone, there is a cell for your skin, there is a cell to develop your brain cells, there is a cell to develop your muscles and heart, because heart is also a muscle, right? There is a cell to develop your intestinal part, there is a cell for each and every part of your body, and these are specialized cells. They do each and every function among themselves. So now you see, what when I said you, there is each and every cell for particular part of your body. Now these are called as specialized cell. A brain cell can only create your brain, and hair cell can only create a hair, and bone cell can only create a bone. But there are particular cells which can create each and every part of our body. It can create an hair, it can create my brain, it can create a heart for me, it can create a skin for me, it can also create a bone for me. That type of cell is called as embryonic stem cells. Embryonic stem cells are those stem cells which have unlimited capability or which can be turned into any cell of the body. Whereas adult stem cells are very specialized stem cells which cannot be turned into any of our body parts, and they are only specialized. For example, if there is a bone stem cell, it can only develop bone stem cells, but it cannot develop the skin stem cells or your skin. The ability of a stem cell to be turned into any part of your body, they are basically embryonic stem cells. Embryonic stem cells, my friend, basically collected from a stage of bryophyte. Bryophytic stage of your embryo formation. If you try to extract these embryonic stem cells, there could be no possibility of child being born. That is, when sperm and egg, they get fertilized. If this is sperm and this is egg, this is a female sexual part and this is a male sexual organism. When they get fused and they start dividing, there is a particular stage before your embryo starts develop into a human shape, that is called bryophyte stage. At this biocyst or bryophytic stage, you can extract the embryonic stem cells. Okay, once you start extracting these embryonic stem cells at this particular stage, you are going to destroy the complete embryo. The complete embryo is destroyed because of this procedure, and there will be no process or there will be no child further. The child will be dead at this stage itself. Okay, so this is something what you have to learn. My friends, because of the technology, as I said you, adult stem cells being specialized cells, very particular cells, they cannot form into any other cell. For example, a skin cell can be formed into only skin cell, bone into only bone, but they cannot be formed into a muscle cell. Now, the technology has developed such a great that we were able to develop an adult induced embryonic stem cells or pluripotent stem cells. That is, even these adult cells could be manipulated or could be developed to become embryonic cells. Can adult cells become embryonic cells? Yes, they can. An adult cell can become an embryonic cell, and we have such technology, and it was able to possible by gene editing. Gene or gene editing. Now, when it comes to genetic therapy, what is this genetic therapy? You all might have a doubt. Okay, gene therapy. Gene therapy. What is this gene therapy? You all have a doubt. Gene therapy is using genetics or genetic technology for the purpose of treating any particular disease. What genetic technology you can use? You can use gene editing. You can also use gene silencing. What is the difference between gene editing and gene silencing? In gene editing, you remove a particular part of your DNA which is basically causing you the disease. Okay, in gene editing what you do? We will cut one particular part of your DNA which is causing you this particular disease, remove it and replace it with a healthy DNA which will make you to live happily. Cut, remove and add an healthy part. That is gene editing. Whereas gene silencing, you are not going to cut anything or remove or add something. We are only silencing that part of your DNA which is responsible for that particular disease. You are going to go into your DNA, such which part of the DNA is causing cancer in your body. I am going to make it silent. I will put some particular enzymes into my DNA. I will say this particular part of DNA is going to get silenced. Okay. So, this is gene silencing and gene editing, great technologies. We have a lot of other gene editing technologies that we will be discussing further. Next, wide biotechnology. This is basically application of biotechnology in the field of industries. For example, Take the pharmaceutical industry or take the food industry. Do you know in food industry we are able to produce large amount of cheese today, right? Cheese. We all eat cheese. How do you think cheese is produced? Okay, cheese is produced when you have curdles. Okay, basically organism which can form curds, lactic acid bacteria. How can you even imagine such a large amount of lactic acid bacteria could be produced? Such a large amount of curdle could be produced. You know, earlier in the times, the baby calves, okay, how could be cheese produced? The baby calves, that is newborn calves, the stomachs of these newborn calves were to be opened and a large syringe used to be inserted into the stomachs, and they used to extract this particular ruminant that was needed to produce this cheese. There used to be a separate part above the stomach, the particular stomach, a ruminant part of their body. From that they used to extract this liquid and use it for this purpose. But the development of biotechnology made it possible to develop this particular ruminant which basically develops in a body of a newly born calf from bacteria itself. Now today we no longer do torture to any of the calves, a small cow or a small baby cow or a small baby buffalo or oxen, whatever it is. Today we use bacteria to develop them. Why don't you think we need yeast to develop the most famous beverages? Yeast, especially beer and alcohol. Yes, we need yeast. Now to improve the flavors of these particular beverages, say your alcohol, or to improve the shell life of a particular food what you are going to develop, or to dissolve all the fibers, or to make fibers digestible. You see, most of the fibers what we eat are not digestible, they come out of your feces. But to make these fibers digestible, okay, all these are possible by using our biotechnology. Okay. So biotechnology, white biotechnology is application of biotechnology in the field of industry. Next, blue biotechnology, it is basically application of biotechnology in the field of water. Okay, that is in the fisheries industry, freshwater organisms and marine water organisms. Next, yellow biotechnology, basically application of biotechnology in the field of insects. Okay. And grey biotechnology, basically biotechnology in the field of environmental degradation or bioremediation processes, that is clean environment. Next, gold biotechnology, this is about bioinformatics. This is about bioinformatics, that is all this knowledge of biotechnology could be taken and interpolated to become a computer program. That is all knowledge we get from experiments, right? We'll make it into a computer program and from then we'll ask our computer to give us solutions. That is artificial intelligence, bioinformatics, decision making, and all those is called as gold biotechnology. So now let's learn about the applications of biotechnology in the field of medicine in particular. My friends, recently we were able to develop a technology called as CART therapy, C-A-R-T therapy. What is this CART therapy? C-A-R-T. CART stands for chimeric antigen receptor T cell therapy. Okay, chimeric antigen receptor T cell therapy. What does this CAR therapy does? Basically, my friends, in your body, there are various defense mechanisms. Your skin is your first defense from any particular disease to enter into your body. Now, even in your blood, there are various types of cells which help you to destroy various types of organisms that are attacking your body or which attack your body to cause a disease, right? And one among those cells is T cells, and these are basically cells which protect your body, protection cells. Now, in typical cancer treatment, what do we use is, we use chemicals called as chemos. What do the chemo do is, a chemo has capability to make your bones porous. An healthy bone, like for example, let's say This is your bone, an healthy bone. A bone which undergoes chemo will become porous, like hole like this. And it is very susceptible for breaking and crumbling. Now what happens is, doctors try to use this chemo to be inserted into your cancer cells, so that even your cancer cells get crumbled like this, and over a period of time they get broken. But this treatment has various side effects, making your bones weaker, feeble, and making yourself also weaker, and there is also a lot of hair loss in this cancer treatment. Now, in order not to go with this treatment and to develop a treatment which has less side effects, we have developed a technology called as chimeric antigen receptor T cell therapy. What do we do in this technology is, we extract the human blood, and from the human blood we extract the T cells. We extract the T cells. Now you see, my friends, your body cells have memory. Your body cells have memory. You might have seen a therapy called as plasma therapy during the time of COVID. What happens in this plasma therapy? If a person is affected by COVID-19 and got cured, say I got COVID-19, I got cured by COVID-19. Now imagine my father or mother got the COVID-19. Now what happens? I, being a person who got COVID and recovered from the COVID, my blood will contain antibodies which can fight COVID-19, right? My blood will contain antibodies which can fight COVID-19. Now what doctors do is, basically they extract my blood and separate the plasma from my blood, in which the antibodies mostly exist, and this plasma will be inserted into my father and mother, so that they get the capability to get recovered or to fight the COVID-19. So this therapy is very famous. In this same way, what doctors do is, they separate your T cell from your blood and take this T cell to the laboratory. They will give T cell a particular memory. See what got? My body cells have got memory from my curing of COVID-19, that my body cells know, if COVID-19 attacks me, what types of proteins that my cells should produce in order to destroy the COVID-19 virus. In the same way, we take a T cell from the blood of the cancer patient, and we will modify this T cell in the laboratory. In the laboratory, what we do? We modify by using gene editing tools, we modify this T cell and insert a particular memory into the T cell, saying that wherever you find this particular type of cancer, go and attack. I am giving a memory input to this T cell, I am training this. Now, this particular T cell which has been trained will be replicated in the laboratory. Will be replicated in the laboratory by using RT-PCR, reverse transcriptase polymerized chain reaction. Basically, not needing of RT, but we will be using a technology called as polymerized chain reaction. This polymerized chain reaction will replicate these T cells, will make into a small bag, and this bag of T cells will be inserted into your body, and these T cells go and attack your cancer cells, and you will be ultimately cured of the cancer. The basic advantage of this chimeric antigen receptor T cell therapy is that these T cells which are modified, being coming from your own body, the T cells which are modified in the laboratory come from the patient itself. Being coming from the patient, this treatment is less invasive and it does not have higher side effects when compared to the all other treatments in the market. This is one example. This is one example how stem cell therapy, how gene editing will help you to treat any of the harmful diseases that exist in the market. My friend, gene editing have also helped you to find cure for the cancer, cure for the HIV AIDS. Even HIV AIDS today is manageable, though complete cure has not been provided. We are managing HIV AIDS just because of this biotechnology and gene editing. Next application in the field of agriculture, I have already discussed. We can develop fortified foods, that is golden rice. We can also develop the genetically modified crops which can give you high yield, high quality and productivity when it compared. And they can also be resistant to various pest attack, and they can also become drought resistant also. Okay. So these are the applications of biotechnology. You can just pause and also read. Next, with respect to animal husbandry, yes, we can increase the yield of a particular breed of a cow to produce more and more milk. Yes, and genetic engineering. You might have seen a movie in Telugu called as Brothers, or it's a Tamil movie of Surya. You can see his father has developed a particular type of a cow breed which will produce a particular type of milk. If that milk has been consumed by those Russian athletes, they will become more and more efficient. So such a type of milks might not be in the society, but you can obviously develop fortified foods, digestible foods, increase the productivity of food, productivity and nutritional capabilities. That is, you can make them therapeutic. You can make them therapeutic foods. So, and you can also do cloning to the animals. You can also develop a particular breed, and you can also develop pigs which can give parts to the human beings. Okay, I will tell you this concept. This concept is known as xenotransplantation. Okay, I will explain you here. Xenotransplantation. Basically, xenotransplantation means taking an organ from animal and inserting that particular organ into human being. That is called as xenotransplantation. Okay, organ transplantation from animal to human is called as xenotransplantation. Recently in USA, a pig heart introduced into human. Now, when I say a pig heart was introduced into a human to save his life, do you think any pig can be used for this xenotransplantation? No. These pigs from which you are extracting any part of the body like kidney, heart or liver are genetically modified pigs which are raised in a closed environment. They are genetically modified so that when this organ is being introduced into your body does not reject this particular part which has been transplanted. That is called as xenotransplantation, and it is only possible by gene editing technologies, not just like that. You know, this biotechnology, when it is applied to animal husbandry, you can develop high yielding breeds. High yielding breeds. You can do artificial insemination. This is also known as selective breeding. Okay. We can also develop clones. Clones are very important, my friend. What is a clone? You seeing yourself. Creating another you from your own cells is called cloning. There is no another female cell. Basically, cloning can be seen in the nature in cases of snakes or any reptiles. This process is called parthenogenesis. What happens in these reptiles is, when reptiles don't get a male partner, okay, when reptiles don't get a male partner, the female reptiles will lay eggs, and these eggs are basically fertilized eggs containing only female based DNA, and there is no male partner. All the offsprings which are born out of these eggs are basically clones from the mother. This can be seen in snakes. That is, they don't need a male partner to lay eggs. Snakes and reptiles don't need a male partner to lay eggs. This process in the nature is called as parthenogenesis. Now, why do we develop clones, my friends? Clones are basically developed to maintain that particular species over a long period of time. Imagine one rhinoceros which is going to be get extinct. In order to save that rhino, we have to develop more clones of this rhino so that you have this rhino for a long period of time, right? So, what is the first clone developed? Basically, sheep was the first clone. Can anyone comment that what is the name of the sheep that was cloned for the first time? Hope you understood what is xenotransplantation also. Next, application in food processing. As I already said you, cheese industry needs lot of bacteria. That particular bacteria, lactic acid bacteria or curdling bacteria, this could be produced in a large extent by using biotechnology. The food can be made more fortified, premium, quality, everything. You can see genetically modified crops I have already discussed. Environment also already been discussed. Okay, and particularly you can also use biotechnology to develop biosensors. Biosensors are nothing but basically they are microorganisms. Whenever they come in contact with a particular pollutant or particular level of pollutant, they react, and that reaction shows you the presence of that particular pollutant in the region. Okay, biosensors. That's the another application. Now let's also learn about the DNA and RNA. This is very important part. Okay, let's start focusing on this. Now, every human or every organism have cells in them, and each and every cell contains some important component called as DNA. In each and every cell of your body, you will find this DNA. DNA is basically a double helical structure with carbon and amino acids and phosphorus bases. Amino acids, phosphorus and carbon bases combined together form this DNA. What does this DNA do? DNA contains entire information of your body when to produce a particular type of protein. See, a growth hormone is a particular type of protein. Your DNA knows when to release this growth hormone. If this particular growth hormone has not been released in time, what happens? You will remain as a short stature person. Your DNA knows each and everything, when to release, what amount to be released. But what part of this DNA releases your that particular protein directly is, that is RNA. Now let's learn about DNA and RNA. DNA stands for deoxyribose nucleic acid. Deoxyribose nucleic acid is basically a double helical structure, you can see here, having two base pairs of amino acids. Those two base pairs include cytosine, guanine, adenine and thymine. This is DNA, right? Cytosine, guanine, adenine and thymine. Here base pairs are basically G and C and A and T. That is, adenine and thymine form one pair all the time and guanine and cytosine form another pair. You can never see a pair coming from adenine and cytosine. So, what are the two base pairs, my friend? You will have adenine and thymine, guanine and cytosine. These are called as base pairs. Okay, these combinations only you can see a DNA. Now let me explain you what is the use of understanding a DNA or your gene. My friend, gene is a small part of DNA. What is gene? When you understand this DNA, gene means this is a small part of DNA. Whereas genome is entire DNA. When I said DNA is based on these base pairs, that is, if you take your DNA, if this is your DNA strand, you can see these base pairs repeat at certain intervals. For example, if it is this AT, 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 and next comes GC, next comes again AT, next comes again GC, next GC. In this way, the base pairs could be arranged in any form. There is no reason or there is no particular pattern that this will be this pattern. That is what comes after base pair of adenine and thymine. Does it only comes guanine and cytosine, or what comes is not predetermined. But this arrangement of these base pairs will tell you whether a particular person has a disease or not. Imagine if I take a sample DNA and arrange their DNA into these base pairs and arrange the DNA into base pairs, and each and every person's total genome. Genome is the entire arrangement of base pairs of DNA has been given, or you have drawn them. If I take the information of 1 crore persons of India, and if I look those 1 crore person and sample out those people in this 1 crore who have diabetes. Imagine there are around 10 lakh people in this 1 crore population affected by diabetes. When I look at the genome of entire this 10 lakhs people, don't you think I will find some pattern in this 10 lakh people which all these 10 lakh people will have common in this arrangement. Some arrangement will be common in this 10 lakh people, and don't you say that this part of the DNA which is very common in all of these 10 lakh people will be the part which is causing diabetes, right or wrong? See, I'll tell you something you might understand. See, let's say adenine thymine, guanine cytosine, adenine thymine, guanine cytosine. So this is one genome. Say this is very short, genome is not this short, genome is very very big. It can go how many kilometers also, like if you write like this. Now, this is genome of one person and this is genome of other person. Let's say GC, GC, AT, AT, AT. This is his genome. Now, can I say this particular part of his genome is both common, and in these two people I can see a diabetes being prominent. Diabetes. And there comes a third person also. Even the third person also has same pairs and he also has diabetes. Can I conclude that this is the part of your DNA which is causing diabetes in your body? And if I am able to remove this damaged part of your DNA and add an healthy part, don't you think I will be cured of diabetes? If imagine if I am able to silence this particular gene, don't you think I will be protected against the diabetes? So, this understanding of DNA and what are those base pairs also makes a lot of sense. Look, remember, in DNA, basically a double helical structure. DNA stands for deoxyribose nucleic acid. DNA has 2 base pairs with 4 amino acids that you can see: cytosine, guanine, adenine and thymine. Whereas RNA, in place of thymine, you can see uracil. In case of thymine, you can see uracil. DNA being very very long in nature, it needs a particular shape to be retained in your cell, because cell cannot accommodate if the DNA is long. Cell is very small. In order the cell to accommodate, DNA takes a form called as chromosomes. What are chromosomes, my friend? In human, there are 23 pairs of chromosomes. What are these chromosomes? Chromosomes are nothing but the DNAs, okay? The DNA molecule being extremely long. As I said, if you can see, it goes long. Hence, without right packing, if you are not packing this DNA properly, it cannot fit into your cell. Thus, DNA are transformed into chromosomes to get accommodated into the cells. As I said DNA has 4 unique base pairs, nitrogenous base pairs: adenine, guanine, cytosine and thymine. Whereas for RNA, in case of thymine, there will be uracil. Next, RNA, deoxyribose nucleic acid. Basically, this is the part of the DNA which directly involves in the synthesis of the protein. This point is very, very important. Basically, this gives message to your cell. What RNA does is directly goes and gives message to your cell saying that please produce this particular type of protein because you are affected by this particular type of disease right now. What happens? DNA contains information. See, DNA is something like book. DNA is something like book. Now, someone has to open this book and study that particular chapter to give a solution or not. Now, who does this? RNA searches this whole book and takes out that particular paper and comes and gives to the cell saying that, Ray, this is the particular paper which you have an answer for your doubt or your exam. See, my cells are being disturbed or affected. My cells need answer to various questions. That is, my cells need answer to resolve a particular disease that I am being affected. So, who gives this information to my cells? This information is being searched in the DNA and being taken and given to that particular cell by the RNA. Okay, RNA has 4 nitrogenous bases: adenine, guanine, cytosine and uracil. Okay, that is very, very important. Now, this is the table which shows you differentiation between DNA and RNA. You can see DNA is deoxyribonucleic acid, whereas RNA is a nucleic acid. Basically, this is very, very important point. Point number 2, DNA can be seen inside the nucleus of a cell and even in some cell organelles. But in plants, it is present in mitochondria. Whereas RNA is basically found in cytoplasm of the cell, but very little it is found in the nucleus. So, RNA can be seen rarely in the nucleus, but DNA is entirely filled in the nucleus. Next, it is double stranded, it is single stranded, it is a long chain nucleotide, it is a short chain nucleotide. Okay, it stores and transfers the genetic information. Over the years and years, what you are gaining because of your traits, that it is stored. And this uses, RNA uses this stored information to be extracted and to be extracted and to be given to the cells to act accordingly. Okay, this has 2 nucleotide strands, this has single nucleotide strands. The base pairs include adenine and thymine, cytosine and guanine. In this case, adenine and uracil, cytosine and guanine are the base pairs. DNA is a self-replicating part of your body. RNA is basically synthesized from DNA. DNA could be destroyed if it is exposed to the ultraviolet rays, and whereas RNA cannot see much damage when it is exposed to the ultraviolet rays. DNA is a long chain polymerizations, whereas RNA is a short chain polymerase, double helical, single helical, and basically DNA is occurring in the form of chromosomes in a human body, whereas these RNAs occurs as ribosomes. What do they occur as? Ribosomes. Okay, the amount of quantity of DNA is fixed in every cell. The quantity of RNA in a cell varies according to whatever you are having. Okay, so this is very important table. Hope you will be able to remember and understand. So this is a small diagram showing you the differentiation between DNA and RNA. When you have learned about DNA, RNA, various types of biotechnology and also the applications of biotechnology, let's now also try to learn few of the other terms with respect to biotechnology. So when I show you this diagram, when you see, this is basically a bacterium. Within the bacterium, you can see a bacterial DNA and a plasmid. My friends, in order to genetic modify a bacteria, it is not easy to modify the bacterial DNA directly. So that's why what we do in genetic engineering is, we will modify that parts of the bacteria which are easy to get modified or easy to get edited. So in the case of bacterium, what we do is, we edit the plasmids. We insert our own traits, whatever we want to insert into the bacteria, those traits are inserted into the plasmids, and by modifying plasmids only, we are able to get our whatever product we want to develop. For example, we develop insulin from E. coli bacteria. What we do is, we genetically modify the plasmid of E. coli bacteria. Coil is genetically modified. Okay, when this E. coli bacterium plasmid is genetically modified, it starts producing insulin. So that's why I have inserted this diagram. And the next point, here you can see one important term called as phage or bacteriophage. What is this term, my friends? Phage or bacteriophage. This one. Now what is this bacteriophage or phage? See, bacteriophage. These are basically viruses which attack. These are basically viruses which attack bacteria. Now what do we do with this bacteriophages is, we develop certain virus, because virus has innate capability to modify our DNA or to disturb our DNA. Because virus might have RNA and DNA. When certain virus enters into your body, they go and attack our DNA. By modifying our DNA, they cause disease in our body. So virus has capability to carry a DNA, or RNA. Basically, virus becomes a carrier of DNA or RNA. Now we use this particular type of viruses, those are called bacteriophage viruses, to insert a particular DNA into a bacterium. What do we use these viruses for? We use this virus to insert a particular type of DNA into a bacteria. So that's why knowing about the bacteriophages is important. What are bacteriophages? They are basically, what are the bacteriophages? They are bacterial viruses which are used for inserting a traits into the bacterium. Next, rDNA technology, my friends. Recombinant DNA technology. One of the most important technology from exam point of view. You know what do we use this rDNA technology? We use this rDNA technology to gene editing. See, look at here. What do we do here is, we select a particular bacterium. In the bacterium, we select a particular plasmid, and within this plasmid, what do we do is, we select a particular part and remove that part, and we will introduce a recombinant part, that is the part or the trait what you want to give to this particular bacteria, and we take this bacterium and insert into a plant cell or develop this bacteria in multiples and develop insulin or various other products. One point you can see here, one very, very important point you can see here, a bacterium DNA could be introduced into a plant DNA. A bacterial DNA or bacterial traits could be introduced into a plant DNA. If a statement comes, the bacterial DNA could be introduced into a plant DNA, yes or no? Yes, the best example for this is GM crops. In that, BT cotton, where BT stands for Bacterium thuringiensis, which is a bacteria. Okay, this is how an rDNA technology works, my friends. Basically, it was developed in the year 1968 by a Swiss microbiologist called as Werner Arber. You can also remember his name if you want. Not much important from the exam point of view. Some other points, these are the steps of basically the rDNA technology. One important point I want to tell is ligation. Ligation is nothing but attaching point. Okay, this is a step of ligation, joining of two pieces which are cut of DNA fragments. Basically, if you are attaching two parts of a DNA, that is called as ligation. This is stitching part or sticking part. Stitching or sticking part in the whole process. Now, where do you apply this recombinant DNA technology? Basically, you can use this recombinant DNA technology to treat HIV/AIDS patients, and it can also be used in diagnostics also. Basically, virus-based diagnostics, that is ELISA test, enzyme-linked immunosorbent assay test. So we can also develop what you can say specialized plants, especially tomatoes like flavsar, BT cotton, basically which is a cotton trait which is protected from bollworm, whereas LSAV and protein rich rice and also vitamin A rich rice can also be provided by using this recombinant DNA technology. So you can see some of the biological products which were developed because of gene editing. We were today able to develop insulin because of gene editing. We are able to produce growth hormone for people who are having pituitary dwarfism, erythropoietin, which is basically developed for anemia. So we are also developing various other components by using gene therapy. Vaccines for various treatments are also produced by gene therapy. Now, one important point, we will try to understand those points which we did not discuss from this part, that is solving a MCQ. Now let's see the question. Recombinant DNA technology allows genes to be transferred across different species of plants? Yes, it is. From animals to plants? Yes, it can. From microorganisms to higher organisms? Yes, it can. So 1 and 2, only this one we haven't discussed, so it might not be possible. From animals to plants we haven't seen or we haven't discussed, but from microorganisms to plants, yes, we can and we will be doing. So option 2 is wrong, 1 and 3 is the right answer in this case. Next, the process of copying genetic information from one strand of DNA into RNA is known as transcription. What is that known as? Taking the information, RNA taking the information from DNA is known as what, as my friend? Transcription. This is very, very important. Try to note this down. Next, which of the following features of DNA makes it uniquely suited to store and transmit genetic information from one generation to another generation? Which part or which structure of DNA is helping it to transfer the genetic information from one generation to another generation? That is complementarity of the two strands. This is the answer. Very, very important. What is the answer? Complementarity of the two strands. Next, which of the following statements about DNA is correct? When you are learning which of the following statements about DNA is correct, then the DNA is hereditary material of all living organisms. No, my friend, DNA along with RNA, DNA and RNA. If it is DNA along with RNA, then this statement could have been right. So if you eliminate option 1, so you can eliminate these two options. Okay. Next, all segments of DNA code to synthesis of proteins. No, not entire DNA will produce proteins, my friends. Part of a DNA will produce proteins. So even this statement is wrong. Next, nuclear DNA is a double helical with two nucleotide chains which run anti-parallel. Yes, obviously option 3 is right. DNA is also found in mitochondria. Yes, we have seen in our notes. Yes, both 3 and 4 is the right answer in this case. Next, we are going to start another topic that is called as gene editing. Gene editing is basically you are going to edit the gene, that is you are going to remove the unwanted strands and you are going to add a new strand. Removing and adding. That is what gene editing happens. Just now you have seen an technology called as recombinant DNA technology. This technology is also a type of gene editing technology. Why do we do gene editing? To remove the unwanted things from particular organism and to improve its traits, right? So for example, to reduce the size of a seed in the mango and increase its flesh. That's what we do for the gene editing, right? So gene editing, they basically target a particular part of your gene, they remove that gene and insert a healthy gene in that. There are various tools of gene editing, my friends, and among all those gene editing, CRISPR-Cas9 is the most cheaper and most efficient tool that one can use. What is this CRISPR-Cas9? CRISPR-Cas9 stands for cluster regularly interspaced short palindromic repeats. What's my friend? Clustered regularly interspaced short palindromic repeats, and Cas9 stands for a protein which basically guides DNA and it helps the DNA to be edited. Okay, I'll show you the diagram how this CRISPR-Cas9 technology works. Here you can see. Now this Cas9 is a protein, my friend. Now this Cas9 goes and attaches to that part of the DNA which you want to remove. Now once it goes and attach, this part of DNA is cut and that cut part is eliminated or removed, and then another healthy part of a DNA is taken and attached. So in the whole process, this Cas9 is playing a very important role. This Cas9 is called as guide RNA. Okay, this is a protein. Cas9 is what? Protein. This is one important example you have to remember. Cas9 is basically a molecular Caesar. Molecular
Caesar.
Nothing else, everything else is written. It's a textbook. You can just go through this. Whatever it is needed, I'll tell you. Like for example, what are the risks associated with gene editing? Because we do not have long-term studies with respect to gene editing. We don't know how an organism will be behaving in a long run once it has been gene edited. We don't know what are the side effects of this gene editing. Sometimes this gene editing could also be used for creating weapons of mass destruction. Weapons of mass biological destruction can also be produced. And basically, Western countries and developed nations having much of this technology, we don't know what they could lead to, and there could also be lot of differentiation among the West and the East with respect to the technology they hold. Now, there is no law in India, my friends, which protects us from gene editing. Basically, Indian IITs, the chimeric antigen receptors T-cell therapy, the CAR-T therapy has been developed by the IITs of India, basically IIT Madras, which is trying to produce solutions with respect to the cancer and HIV/AIDS treatment. Let's see some of the important questions. Scientists can cut apart and paste together DNA molecules as, you can see this question, very beautiful question. It is saying statement, scientists can cut apart and paste together DNA molecules at will regardless of the stores of the molecules. Yes, they can do it because of gene editing. DNA fragments can be manipulated using restriction endonuclease and DNA ligases. Yes, absolutely correct. Both A and R are individually true, and R explains A very correctly. Okay, so option A is very much correct, my friends. Next, CRISPR-Cas9, the Cas9 protein often mentioned. What is it? It's basically a molecular Caesar. They are basically, Cas9 is a protein which helps you to cut the DNA. They are called as molecular Caesars. With reference to the recent development in the science, which one of the following statements is not correct? Very important. Functional chromosomes can be created by joining segments of DNA taken from the cells of different species. Pieces of artificial functional DNA can be created in the laboratories. A piece of DNA taken out from the animal cell can be made to replicate outside the living cell in a laboratory. Cells taken out from the plants and animals can be made to undergo cell division in the laboratory in the Petri cells. This is obviously possible because in tissue culture that we can do. Now let's see what are the other thing. A piece of DNA taken out from an animal cell can be made to replicate outside the living cell in a laboratory. Yes, but is it outside the living cell? Yes, some nutrients are to be given, they can be replicated. Basically, polymerized chain reaction is what that we do today. So this is also correct. Let's see other one. Pieces of artificial functional DNA can be created in laboratories. Artificial functional DNAs cannot be created in the laboratories, my friend. This is wrong. All the parts of the question are absolutely correct. Now, as I said you, there is a technology called as gene silencing technology. When compared to gene editing, what is gene silencing means? You are silencing that particular part of your DNA which is causing you disease. That is, you are making or switching off this particular part of your DNA. Okay. What you do is basically this DNA has some information in it, and this information will be going into the RNA, that is called as transcription. Now RNA uses a process called as translation. These two terms are important, please note down. Transcription is extracting the information from DNA by RNA. Now translation is RNA giving information to the cell to produce a protein. RNA to protein is called as translation. DNA to RNA is called as transcription. Okay. Now what we do is, we do not allow this RNA to undergo translation. So that's why you will not produce this protein which is responsible for disease. Okay, we are stopping RNA to turn into translation and to produce that particular proteins which are causing diseases. So there is no translation stage, and this type of technology is called as gene silencing. Remember, transcription and translation. Transcription is extracting the information from the DNA, whereas translation is turning this information into a protein by the RNA. Now, if you are not able to produce this particular protein which is causing the disease, then you are also protecting yourself, right? That is called as gene silencing. Next, RNA interface technology, my friends. This is also another type of gene editing technology that we have developed in these days. This technology is basically used in developing cure for HIV AIDS. In gene therapy, as I said you, what happens in gene therapy, my friends? One, that is gene silencing. You can silence the disease causing gene, or gene editing, that is you can remove the disease causing gene and add a healthy gene. That's what, replacing the disease or inactivating the disease causing one. Okay, this is a typical treatment of gene therapy, in vivo and ex vivo. When the treatment happens within the body, that is called in vivo. Ex vivo is treatment happens outside the body. For example, you can see this is CAR therapy. For example, I am taking the T cells from my body, I am modifying them in the laboratory, I am replicating them in the laboratory and again inserting them into the human body, right? Chimeric antigen receptor therapy happens like this. Or if I use a bacteriophage like a viral which can carry a DNA, and if this virus is allowed to be attacked into my body, once this virus enters into my body, it starts modifying my DNA through the DNA which is inserted into this particular virus, and this virus has been created by me in the laboratory. Now virus treats my body in such a way. Okay, so this is also another technology, what you can say. In vivo, that is treatment completely happening inside my body through gene editing using viruses. Ex vivo is that my body cells are extracted, they are edited in the laboratory and they are reinserted into my body by using various sources. Next, there are different types of gene therapy. These are very important points, these two, hope you can note them down. One is somatic gene therapy. That is, effects will not be transferred to the next generation. If I am undergoing a gene therapy, my children will not have the same traits that I have gained because of gene therapy. That is called as somatic gene therapy. That is, my next generation will not be protected because of my gene treatment. But there are certain gene treatments that if I gain a particular trait, this trait will also be transferred to my next generation, that is called as germline gene therapy. For example, if I am treated for anti-cancer, then my children will also be anti-cancer protectants, that is they don't get any cancer. If I am treated for diabetes, my children will also be not having any diabetes. That is called as germline gene therapy. Whereas somatic gene therapy is, if I am treated, my children might or might not be affected. That's what it is. These two are very important. Next, three parent baby or mitochondrial gene therapy. Very important, my friend. My friend, mitochondria is the powerhouse of the cell. But in certain mothers, the mitochondria is so weak that they cannot give birth to their children. But they have a nucleus which is very active. Okay, see, this is mitochondria and this is a nucleus. Okay, nucleus can survive only because of the mitochondria around it. Now, when this mitochondria, you know, we have seen nucleus contains the DNA, mitochondria also contains the DNA, both of them contains the DNA. Now, if a mother is having weak mitochondria, what we do is, we select a donor mother, we select another mother and we take the nucleus of this mother who wants to have a baby. I'll write, this mother wants to have a baby. We take the nucleus of this mother and insert that nucleus into the mitochondria of another mother. Now what happens, this mitochondria will allow this nucleus to survive, and when this is basically an egg, when this egg is fertilized by the sperm of the father, who is the father, then you can form a baby. How many people are there in this process to form one single baby? One mother from whom you have taken a nucleus, another mother from whom you have taken a mitochondria, a father from whom you have taken a sperm. So this process is called as three baby or three parent baby. Okay, three parent baby. You can see a damaged mitochondria in the mother's cells. So our nucleus is extracted and being inserted into a healthy woman mitochondria, and this egg is being fertilized by using the sperm of the father, and this causes a phenomena called as three parent baby. Okay, and this transfer of nucleus, see, you have transferred the nucleus of one mother into the another mother, right? This transfer of nucleus is considered as pro-nucleus transfer technology. Why do we use pro-nucleus transfer technology, my friends? Let's read the options. Fertilization of eggs in vitro by donor sperm, genetic modification of the sperm cells, development of stem cells into functional embryonic stem cells, prevention of mitochondrial disease in the offspring. This is basically to prevent mitochondrial diseases in the offspring. That is very, very important. Next, the term spindle nuclear transfer. Pro-nuclear transfer, in which case do we use these two terms, spindle nuclear transfer and pro-nuclear transfer? Basically we use it in the three parent baby IVF technologies, in vitro fertilization technologies. These two are basically previous year questions. Next, genome sequencing. What is this genome? As I said your DNA has lot of base pairs arranged in a long line. For you to study this entire line and to get the picture of this entire DNA is called as genome. What is genome? Your entire DNA being studied is called as genome. Why do we need to study genome? In order to predict the diseases, in order to save you from harmful diseases, and also to build a profile for the pharmaceutical industries to develop products which are suitable for your particular DNA. My friends, today most of the medicines that we use are Western developed. Do you think that Western DNA and our Indian DNA both are same? No, right? In order to create a profile of India, we have started a project known as Genome Project. Here you can see the difference between gene and a genome. Gene is a small part of DNA, whereas genome is the entire DNA itself. If you are drawing the entire DNA, that DNA is called as genome. Whereas gene is a hereditary element, all the set of nuclear DNA is your genome. So if you are studying the entire genome, you can understand which parts of your genome are causing your particular disease. So India has been able to start a project called as India Genome Project. We were able to sequence a human genome. Before India, USA has already started in the year long back in 2003, and they were able to form their own genome. So under Genome India project, this Genome India project falls under the Department of Biotechnology, started in the year 3rd January 2020. It has collected almost 10,000 different samples from all across India, and we were able to create our own genome, and the project has been successfully completed. Various institutions like Indian Institute of Science, Bengaluru, and IITs, and even Center for Brain Research, Bengaluru, has collaborated in making this project successful. So Indigen project, this is also basically making a genome for India. This was a project which was taken up by CSIR in 2019, not much important, but remember Genome India project, that's what it is important. So to develop bioinformatics or to develop medicines, to develop the genome structure for the future India, we need it. So as I said you, bioinformatics is basically application of biotechnology along with information technology. What are the applications? We can look into the genome and compare our genome with a reference genome. See, what will happen is, we have taken the sample of 10,000 people with various profiles. Now we have a reference genome, a reference page. Now with this reference page, I will compare my page. Now by comparison, what will I get? I will get what type of diseases I could be affected from. Okay, what could be the best possible treatment for me from all this information. So this bioinformatics, what it does is, it does this comparison in a very faster way. It helps you to develop a particular treatment in a faster way, tailor-made treatment, a tailor-made medicine. What is particular medicine that you have to be given in order to treat you? Those are here. Those are here. Those are the advantages of bioinformatics, my friend. And India being the world's first country to have bioinformatics, even before USA also. That is one point I want you to remember. India was the first country in the world in the year 1987 to establish bioinformatics or biotechnology information system, when no other country has this bioinformatics. Okay, Department of Biotechnology has been looking into this bioinformatics field since long. Let's solve one last question. With reference to the agriculture in India, how can the technique of genome sequencing, often seen in the news, be used in the immediate future? What is the future of this genome sequencing? See, genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance in the crops. Yes, you can. This technique helps in reducing the time required for the development of new varieties of crops. Yes, once you have a reference genome, you will have less time. You need less time to solve the solution. It can be used to decipher the host pathogen relationship in the crops. Yes, it can also be used for that. So all the statements are correct with respect to the genome sequencing. In the context of developments in the bioinformatics, the term transcriptome stands for. Transcriptome stands for the description of mechanism of gene expression, a mechanism of genetic mutations, the full range of mRNA molecules expressed by the organism, a range of enzymes used in the genome editing. In bioinformatics, transcription stands for the description of mechanism of gene expression. Now let's look into the environmental biotechnology part in the biotechnology. Environmental biotechnology is basically using biotechnology to reduce environmental pollutants and also to study the environmental pollutants. Now when you see into the application of biotechnology in environmental pollution, you have to understand few terms. Number one, bioremediation. Bioremediation is using biological components to reduce the pollutant effects. That is, you can use microorganisms, basically bacteria, to clean the pollutants. Example, your soil might be polluted. Now in order for your soil to be clean, you use microorganisms in that soil. These microorganisms eat away all the harmful pollutants in that soil and make that soil clean. Or they can also make the most harmful components of your pollutants into the less harmful component of your pollutant. That is, they reduce the most harmful into less harmful parts. That is called as bioremediation. My friends, bioremediation is a process which can be used in treating of polluted water also. What do we do? In the polluted water, we put bacterium, we supply it with enough amount of oxygen. See, we supply enough amount of oxygen and microbes, and these microorganisms will eat away all the pollutants in the water and make the water clean and clear. This process is called as what, my friends? Bioremediation. Okay, the next one, in situ bioremediation and ex situ bioremediation. My friend, bioremediation also has different types. One is in situ and another is ex situ bioremediation. In situ means within the site itself. Where the pollution happened, there only I will give you the treatment. That is called as in situ bioremediation. Example is bioventing. In bioventing, what do we do is, we drill all the holes into the soil. Okay, and we push the air into the soil so that all the microorganisms living in the soil are get activated, and that activated microorganism will start eating your pollutant and make it clean and clear. Okay, supply of air and nutrients through the wells to the contaminated soil to what? Activate or stimulate the growth of the bacteria which can eat away this pollutant. That is called as bioventing. Next, biosparging. In biosparging, what we do is, we inject air into the groundwater. This is biosparging. We are injecting air into the groundwater, and this pressure which you are putting into the groundwater, this starts the biological activity in the groundwater. Now these microorganisms living in this groundwater become activated and they entirely eat away the pollutants in the water. That is called as biosparging. Next is bioaugmentation. This is basically microorganisms are developed somewhere. Genetically modified microorganisms developed in the laboratory are brought to the site and spread in the site. In bioaugmentation, what happened? You are bringing bacteria from outside or microorganisms from outside and you are spreading that microorganism in the site. And slowly these microorganisms eat away all the pollutants and your area will become clean. For example, oil zapper, a bacteria which is developed by TERI, the Energy and Research Institute, can use for clearing the oil spills. What is an example? Oil zapper. Next, exitive bioremediation. That is, you take all this polluted soil, all the polluted material and take it to a particular reactor or take it to a particular scientific location where you will be treating this polluted material. For example, land farming. What do we do is, we extract all this land which has been polluted, take it to a place and we will expose this land to a good air water mixture, then this land becomes clean and clear. That is called as land farming. Example is this. You take the soil which is polluted, you spread that soil on the land, you put water, nutrients and everything and you make this soil fertile once again. Bioreactors. In bioreactors you will see a reactor like this. A big cylinders in which you will put water or soil and you will also put microorganisms and you provide a conducive environment and make them pollutants lesser. Next is composting. In composting, what you use? You grow the microorganism in the soil which eat away all your harmful pollutants. Next is biopiles. In biopiles, what you do is you maintain a big pile. All this soil which is polluted, all this soil is made into a pile like this. This pile is covered by a sheet and to this pile you will keep on sending microorganisms, air, water so that all the pollutants are cleared. You will give good amount of air. Mycoremediation. Now you see, in bioaugmentation or bioremediation, if you are using microorganisms, it is called as bioaugmentation. You see, bioaugmentation, you use microorganisms. Whereas mycoremediation, this is also a type of environmental cleaning, but here the organism that you are using will be fungus. Basically you use mushrooms for the treatment of your water or pollutants, and these mushrooms can also be genetically modified such a way that these mushrooms will be absorbing all the heavy metals or all the pollutants which are available in the soil through its roots. This will be observing like a sock or a sponge all the pollutants from the ground, and it can also be used for treating of water. They are basically natural sponges which can treat water and can also treat soil pollution. They help in treating your heavy metal pollution also. Microfiltration, this you can see. These are the spores of fungus spores. I will put those fungus spores into a medium, basically this is a husk. Now when I put them into the husk and spray water, husk will provide a warm base and this water will start growing the fungus, and this grown fungus can be put at nalas where this nala water will be thrown through this fungus activated paddy husk, and all that water will be cleaned once it passes through them. Bioremediation by fungus. You can see these are the various types of fungus and what are the toxic chemicals these fungus can eat away. You cannot mug up this, but remember that fungus can also be used in observing heavy pollutants or chemicals from the water and the soil. They can also be used in heavy metal extraction also. You can see that. Heavy metals, basically they cause something called as you have seen in your environment, biomagnification and bioaccumulation. These heavy metals can go into your fat. They become accumulated in your fat, and over the years they become higher and higher in your body, and that will cause a big trouble for you, and that can also be reduced because of controlling those things. You can see bacteria can also be used for observing the heavy metal pollutants in the bioremediation technique. You can see Bacillus sp can observe copper and zinc type of heavy metals. You can see Aeromonas sp can observe the chromium in the pollutants, and you can see citrobacter can observe cadmium, lead and uranium pollution also from the surrounding areas. This is another point you have to note down. Even bacteria can be used to observe the heavy metals and harmful toxic chemicals. This is typically examples what I am giving you. Even algae can also be used. Algae, which is basically a floating material on the surface of the water, to observe the heavy metals and harmful chemicals also. So this is basically a diagram. What are the different types of microorganisms you can use to reduce your pollutants in your soil, water, etc. So genetic engineering will help you to develop such type of microorganisms, or sometimes even the plants. You can see when you are using the plants to reduce the pollutants in the soil, water and in the surrounding, that type of technology is called as phytoremediation. What is that? Phyto. And genetic engineering will help you to develop these microorganisms and these particular types of plants to reduce the pollutants. You can see, let's do this one question. In the context of solving pollution problems, what is or are the advantage or advantages of bioremediation technique? What is bioremediation? Bringing the microorganisms to reduce the pollutants. It is a technique for cleaning up pollutants by enhancing the same biodegradation process that occurs in the nature. Yes, you are using the same thing. Any contamination with heavy metals such as cadmium, lead can be readily and completely treated by bioremediation using microorganisms. Complete treatment is not possible. Partial treatment is obviously possible. So you cannot go for the complete. Genetic engineering can be used to create microorganisms specifically be designed for bioremediation. Yes, you can utilize for them. Answer will be option C, 1 and 3. Next, the process of using microbes to treat areas of land or sea that has been contaminated by pesticides, oil or solvents is known as. What is that, my friend? Bioremediation technique. What is the technique is called as, my friend? Bioremediation. So this is your bioremediation. Next is phytoremediation. That is, you have developed a genetically modified crop or a modified plant which can absorb all the pollutants in your soil. And these crops, when they are put into this contaminated soil, imagine there is a contaminated field. I will grow these plants in that contaminated field. These plants slowly absorb all the pollutants, and at once I will extract these plants. I will pluck these plants and put them into the incinerator so that all the pollutants in the soil are dead. It can use for removing heavy metals, okay, and etc. When you are doing phytoremediation, there are various processes in treating the pollutants. One is phytoextraction. In phytoextraction, what happens? This plant will absorb all the harmful pollutants from the soil. That is phytoextraction. You are extracting the pollutants. Next is phytostabilization. That is restricting this particular pollutant from moving this place to another place. You are not allowing this pollutant to go further into water or any other source. So that is called as phytostabilization. The plants can do that. Next is rhizodegradation. That is, by using this particular plant, you will break the harmful pollutants into less harmful pollutants. What is happening here? In rhizodegradation, the most harmful pollutants are reduced into less harmful pollutants. That is called as rhizodegradation. Next, rhizofiltration. More toxic chemicals are absorbed away. Photovolatilization. Phytovolatilization is nothing but, see, harmful chemicals are absorbed by the plant roots, and over they will be broken into less harmful and they will be released into the atmosphere once again. Now if you are releasing less harmful materials into the atmosphere, that is called as phytovolatilization. Hope you understood what is phytovolatilization. Advantages: they can be easily removed. Plants are greenery in nature, they will also help you naturally, they will improve the fertility. Disadvantages is basically you don't know how to treat with these plants. So that's why. Some plants which can be used for phytoremediation include mustard, avalu we call in Telugu, willow, the plant from which you make your cricket bats, poplar tree, Indian grass and sunflower all help in phytoremediation. So be careful once you are having your sunflower oil seeds, because they can cause heavy metal pollutants in humans also. Now whereas biosensors are basically using microorganisms and biological components to determine the presence of a pollutant. So that's why they are called as biosensors. And bioenzymes, as we know, what are enzymes, my friend? Remember this point, enzymes are proteins. They help in speeding up the biological activity and speeding up the process of removing the toxins from the surrounding. Rhizosphere biodegradation. What is rhizosphere biodegradation? What we'll be using? Rhizo means fungus. You will be using what? Fungus. Basically microorganisms. Relationship between the plants and microorganisms is rhizosphere biodegradation. So after this, we'll also see one more point with respect to the what is stem cell therapy and what are the different types in stem cells. That also is very important. Now read one more question. Which phytoremediation technique uses plant roots to take up contaminated groundwater? What is that? Contaminated groundwater or the surface water. What is happening? Plant roots are taking up. Taking up in the sense, is it phytodegradation or phytovolatilization or rhizofiltration or phytostabilization? In phytostabilization, what happens? It is holding it there itself. In volatilization, what happens? It absorbs and throws into the atmosphere less harmful pollutants. Phytodegradation, you will be breaking down the harmful chemicals into the less harmful chemicals. Rhizofiltration is what here it is. Answer is rhizofiltration. We will see one more important part in this biotechnology, that is stem cell therapy. Let's see what is important in the stem cell therapy. As I already discussed, stem cells are basically specialized cells, and there are few stem cells which are not specialized. Those are embryonic stem cells. They can be converted into any cell. They can be converted into any cell. See, each part of your body, your hair has a hair stem cell. Okay, your skin has a skin stem cells. Your heart has a muscle stem cells. So each part of your body has a specialized stem cells, right? Now some of these stem cells can be used for treatment such as cancer. I have already discussed with you. I am here to just show you some of the figures that which are very important. As I said you, this one, embryonic stem cells, these are extracted at this stage. You can see before the baby is being formed, you will extract these stem cells from a part called this blastula. This blastula will be broken, and from this you will extract this embryonic stem cells. You use these embryonic stem cells for lot amount of treatment. You can use in the treatment for gaining the vision. You can use treatment for brain cell diseases. Okay, you can use this treatment for genetic diseases also. Blastocyst, this is what I have already said you. At the stage of blastocyst stage, these cells are broken. There are different types of stem cells. They are multipotent stem cells, that is stem cells which can produce different other cells. Unipotent, that is one stem cell can produce only one particular type of component. Adult stem cells, they are specialized stem cells. For example, if you have a liver cell, there is only liver what you can produce from it. But even these adult cells can also be transformed into embryonic stem cells, that is induced pluripotent stem cells. You can make these adult cells into pluripotent stem cells, that is by using genetic engineering. These are different stem cell therapies that I just wanted to show you or showcase you so that you can look at them. Basically in India there is no particular law dealing with the stem cell therapy, my friends, but stem cells are very important for treating various diseases, and they can also be used in studying various effectiveness of drugs. They can be used in vaccination, they can be used in development of new drugs, testing of new drugs also. But let me show you one question. Stem cells can be derived only from the mammals. No, stem cells can be derived from any of the animal, not just from the mammals. Stem cells can be used for screening of new drugs. Yes, obviously. Stem cells can be used for medical therapies. Yes, obviously. So the answer C, that is 2 and 3, is absolutely correct with respect to this question. Let me show you one more question. With reference to the latest development in the stem cell therapy, consider the following. The only source of human stem cells are the embryos at the blastocyst. No, they are the source of embryonic stem cells, not for the entire human stem cells. At any stage you can extract the human stem cells. The stem cells can be derived without causing destruction to the blastocyst? No. As I said you, without causing effect to the blastocyst, no one can extract stem cells. So option 2 is absolutely wrong. So these two statements are wrong. 3 and 4 or 1, 3 and 4 should be taken. So this shows that 3 and 4 are right. That is, stem cells can regenerate themselves in vitro virtually forever. Indian research centers also created a few cell lines which can be developed into many types of tissues. Yes, these two are right. And 1 being absolutely correct. The only source of human stem cells is not your blastocyst. From other sources also you can get your stem cells. So answer will be option 3 and 4. So here we come to the end of your biotechnology. Hope I was able to make you informed with various types of technologies in the biotechnology. Though I was able to skip because it being a revision program, we will be discussing each and every part of this in detail in our classes in the later sessions. So this is the end of the session, my friends, of biotechnology. Thank you everyone.